हम लोगों ने क्लास 11 की सारी चैप्टर्स एनसीईआरटी रीडिंग ख़त्म कर ली है। अब हम लोग क्लास 12 की चैप्टर्स को एनसीईआरटी रीडिंग स्टार्ट करेंगे और जैसे ही एनसीईआरटी रीडिंग ख़त्म होती है इसके बाद हम लोग एक एक करके सारे लेक्चर्स को लाएंगे, सारे चैप्टर्स के सारे लेक्चर्स को लाएंगे। ओके, सो टुडेज क्लास 12 चैप्टर 1 सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट्स। लेट गेट स्टार्टेड। Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness, but also all flower plants show sexual reproduction. A look at the diversity of structures of the inflorescence, flowers, and floral parts shows an amazing range of adaptation to ensure formation of end products of the sexual reproduction, the fruits and the seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants, that is angiosperms. Flower, a fascinating organ of the angiosperm. Human beings have had an intimate relationship with the flower since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural values. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. etc. List at least 5 flowers of the ornamentals value. That is your activity based questions, as you can do it in the rest time. To a biologist, flowers are morphological and embryological marvels at the sites of the sexual reproduction. In earlier classes, you have read the various parts of a flower. Figure 1.1 will help you recall the parts of a typical flower. Can you name the two parts in a flower in which the two most important units of sexual reproduction develop? Now, pre-fertilization structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structures, the endoecium and the gynoecium, differentiate and develop. You would recollect that the endoecium consists of a whole of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. So the stamen, microsporangium and pollen grain. Figure 1.21 shows that the two parts of the typical stamen, the long and the slender stalk called filament and the terminal generally bilobed structures called anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and the length of the stamen are variable in flowers of different species. If you were to collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagram would elucidate the range in the shape and the attachment of the anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are dithecous. Often a longitudinal groups run lengthwise, separating the theca. Let us understand the various type of tissue and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. The microsporangia develops further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structures of microsporangium in a transverse section. A typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers: the epidermis, endothecium, middle layer, and the tapetum. The outer 3 wall layer perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grain. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. So, can you think how tapetal cells become binucleated? When an anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. What would be the polarity of the cells of the tetrad? That's a question. So NCRT is loading. As each cell of the microscope. So let's get read from the beginning. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is potential pollen or microspore mother cells. The process of formation of microspores from a pollen mother cells through meiosis is called microsporogenesis. The microspore as they are formed are arranged in a cluster of 4 cells. The microspore tetrad, as the anthers mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Pollen grains. The pollen grains represent the male gametophyte. If you have touched the open anthers of hibiscus or any other flowers, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, size, shapes, colors, design seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 20 to 50 micrometers in a diameter. It has a prominent two-layered wall. The hard outer layer, called exine, is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grains exine has prominent aperture called germ pores, where sporopollenin is absent. Pollen grains are well preserved as fossils because of presence of sporopollenin. The exine exhibits a fascinating array of patterns and design. The inner wall of the pollen grains are called intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grains is surrounded by a plasma membrane. When the pollen grain is mature, it contains 2 cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has an abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of the angiosperm, the pollen grains are shed at the 2-celled stage. In the remaining species, the generative cell divides mitotically to give rise to the 2 male gametes before pollen grains are shed, 3-celled stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders as like asthma, bronchitis, etc. etc. It may be mentioned that Parthenium or carrot grass that came into the India as a contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption have been claimed to increase the performance of athletes and race horses. When once they are shed, pollen grains have to be laid on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remains viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminaceae and Solanaceae, they maintain viability for months. You may have heard of storing semen sperm of many animals including humans for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen -196 degrees Celsius. Such stored pollens can be used as pollen banks similar to seed banks in crop breeding programs. The pistil, megasporangium and embryossac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistils may be fused together, syncarpous, or may be free, apocarpous. Each pistil has 3 parts. The the stigma, style, and the ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated cylinder part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, locule, and the placenta is located inside the ovarian cavity. Recall the definition and the type of placenta that you have studied in class 11th. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1 with paddy mango to many papaya, watermelon, orchids. The megasporangium ovule. Let us familiarize ourselves with the structures of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovules fuses with the funicle in a region called hilum. Thus, hilum represents the junction between the ovule and the funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircles the nucleus, except at the tip where a small opening called micropyle is organized. Opposite the micropylar end is the chalaza representing the basal part of the ovule. Enclosed within the integument is a mass of the cells called nucellus. It's not nucleus, it's a nucellus. Cells of the nucellus have abundant reserve food material. Located in the nucellus is a embryo sac or a female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cells in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus, and MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis resulting in the production of 4 megaspores. Female gametophyte. In majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develops into a female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. So there is a question which you can deal. Let us study about the formation of the embryo sac in details. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to the opposite poles, forming the 2 nucleate embryo sac. The 2 more sequential mitotic nuclear division results in the formation of the 4 nucleate and the later 8 nucleate stage of the embryo sac. It is of the interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed by immediately by cell wall formation. After the 8 nucleate stage, the cell wall are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observes the distribution of cells inside the embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. The 3 cells of the group together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have a special cellular thickening at the micropylar tip called filiform apparatus, which play an important role in the guiding the pollen tubes into the synergids. 3 cells are at the chalazal end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at the maturity through 8 nucleate is 7-celled. Now, pollination. It is in the preceding chapter sections you have learned that male and female gametes in the flowering plants are produced in the pollen grains and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Now this is a question. Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to a stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? So here also a question. Kinds of pollination depending on the source of pollinating. Pollination can be divided in 3 types. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to a stigma of the same flower. In the normal flower, which opens and exposes the anthers and stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity, and also the anthers and the stigma should lie close to each other so that self-pollination can occur. Some plants such as Viola, oxalis and Camelina produce two types of flowers: chasmogamous flower, which are similar to flower of the same other species with exposed anthers and stigma; cleistogamous flowers which do not open at all. In such flowers, the anthers and stigma lies close to each other. When anthers dies in the flower buds, pollen grains come in contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous, as there is no chance of cross pollination landing on the stigma. Cleistogamous flowers produce assured seed weight set even in the dehiscence of pollinators. Do you think that cleistogamy is advantageous and disadvantageous to the plant, and why? Now, zootonogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Although zootonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant. Xenogamy, transfer of pollen grains from anther to the stigma of different plant. This is only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Now, agents of pollination. Plants use two abiotic wind and water and one biotic animals agent to achieve pollination. Majority of the plants use biotic agent for the pollination. Only a small portion of the plant use abiotic agents. Pollen grains coming contact with the stigma is a chance factor in both wind and water pollination. To compensate for this uncertainties and associated loss of pollen grains, the flower produces enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. Wind pollination also requires that pollen grains are light and non-sticky so that they can be transported in wind current. They often possess well-exposed stamens so that pollens are easily dispersed into the wind currents. A large, often feathery stigma is to easily trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into the inflorescence, and a familiar example is the corncob. The tassels you are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledonous. As against this, you will recall that water is a regular mode of transport for the male gamete among the lower plants groups such as algae, bryophytes and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of need for water for the transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria and Hadrilla, which grow in fresh water, and several marine grasses such as Zoastera. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insect or wind, as in most of the land plants. In Vallisneria, the female flowers reach the surface by water by the long stalk, and the male flowers or pollen grains are released onto the surface of water. They are carried possibly by water currents. Some of them eventually reach the female flowers and the stigma. In another group of water pollinated plants such as cigar grasses, female flowers remain submerged in water and pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried possibly inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by a mucilaginous covering. Both the wind and water pollinated flowers are not very colorful and do not produce nectar. So there is a question, what would be the reason for this? So majority of the flowering plants use a range of animals as pollinating agents: bees, butterflies, flies, beetle, wasp, ants, moths, birds, sunbirds and hummingbirds, and the bats are the common pollinating agents. Among the animals, insect, particular bees, are the dominant biotic pollinating agent. Even larger animals such as some primates, lemurs, arboreal, tree-dwelling rodents, or even reptiles, gecko lizard, and garden lizards have also been reported as a pollinators in some species. Often, flowers of animals pollinated plants are specifically adapted for a particular species of animals. Majority of insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into inflorescence to make them conspicuous. Animals are attracted to flowers by color and or fragrance. The flowers pollinated flies and beetles secrete foul odors to attract these animals. To sustain animal visits, the flowers have to provide rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the animal visitor comes in contact with the anthers to and the stigma. The flower body of the animals gets coated of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about the pollination. In some species, floral rewards are in providing safe places to lay eggs. As an example is that of the tallest flower of Amorphellus. The flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant yucca, where both species, moth and the plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larva of the moth comes out of the eggs as the seed start developing. Why don't you observe some flowers of the following plants or any other available to you? Cucumber, mango, pipal, coriander, papaya, onion, lobelia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out. There is a question and they could be pollinated. You will have to patiently observe and flowers of a few days and at the different times of the day. You can also try to see whether is any correlation and characteristic of flowers to animals that visit. Carefully observe anything. Such visitors are referred to as pollen nectar rovers. You may or may not be able to identify pollinators, but you are surely enjoy your efforts. Outbreeding devices. Majority of the flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination result in the inbreeding depression. Flowers plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma respectively are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy, and the third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollination from the same flower or the other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and the female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not heterogamy. In several species such as papaya, male and female flowers are present on different plants that each plant is either male or female dioic. This condition prevents the both autogamy and heterogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species has the same stigma. Often pollen of the wrong type, either from the other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type compatible or of the wrong type incompatible. If it is of the right type, the pistil accepts the pollens and promotes the post-pollination events that leads to the fertilization. If the pollen is of the wrong type, the pistil rejects the pollens by preventing pollen germination on the stigma of the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or the rejection, is the result of the continuous dialogue between the pollen grains and the pistil. The dialogue is mediated by the chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interaction leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatible device pollination and pollen grain germinates on the stigma to produce a pollen tube through one of the germ pore. The contents of the pollen grains move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. You would recall that in some plants, pollen grains are shed at two-celled condition, a vegetative cell and generative cell. In such plant, generative cell divides and forms the two male gametes during the growth of the pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. Pollen tubes, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Many recent studies have shown the filiform apparatus present at the micropyle part of the synergist guides the entry of the pollen tube. All these events comes from the pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as the pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or emission of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction. Even in incompatible pollination to get desired hybrids, you can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalaria, balsam and vinca on a glass slide containing a drop of sugar solution about 10%. After about 50 to 30 minutes, observe slide under the low power lens and microscope. You are likely pollen tubes coming out from the pollen grains. A breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approaches of the crop improvement program. In such crossing experiments, it is most important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted plants pollen. This is achieved by emasculation and bagging techniques. If the female parents bear bisexual flowers, removal of the anthers from the flowers bud before the anther dehiscence using pairs of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flowers attains receptivity, mature pollen grains collected from anthers of the male parent are dusted on the stigma and the flowers are rebagged and the fruits allowed to develop. If the female parent produce unisexual flowers, there is no need for the emasculation. The female flowers buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out by using the desired pollen and the flower rebagged. Now, double fertilization. After entering one of the synergists, the pollen tube releases the two male gametes into the cytoplasm of the synergist. One of the male gametes moves towards the egg cells and fuses with its nucleus and thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other mother gametes towards the two polar nuclei located in the central cell and fuse with them to produce a triploid primary endosperm nucleus, and as this involves the fusion of three haploid nuclei, it is termed as triple fusion. Since two types of fusion, syngamy and triple fusion, takes place in an embryo sac, the phenomenon is termed as double fertilization, an event unique to flower plants. The central cell plant triple fusion becomes a primary endosperm cell and develops into the endosperm, while zygote develops into an embryo. Post-fertilization structures and events. Following double fertilization events of the endosperm and embryo development, maturation of ovules into seed, ovary into the fruits are collectively termed as post-fertilization events. Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and form a triploid endosperm tissue. The cells of this tissue are filled with the reserve food materials and are used for the nutrition of the developing embryo. In most common types of endosperm development, the primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, the cell wall formation occurs and the endosperm becomes unicellular, and the numbers of free nuclei formed before the cellularization varies greatly. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and surrounding white kernel is called cellular endosperm. Endosperm may either be completely consumed by the developing embryo, pea, groundnut, beans before seed maturation, or it may persist in the mature seed, example castor or coconut, be used up in the during seed germination. Split open some seeds of castor, peas, beans, groundnut, fruits of coconut, and looks for the endosperm in each case. Find out whether endosperm persistent in cereals, white rice, and maize. Now embryo. Embryo develops at the micropylor end of the embryo sac where the zygote is situated. Most zygotes divides only after certain amount of endosperm is formed. There is adaptation to provide assured nutrition to the developing embryo through the seed differ greatly. The early stage of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure depicts the stage of embryogeny. In a dicotyledonous embryo, the zygote gives rise to the proembryo and subsequently to the globular heart-shaped and matured embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of the embryonal axis above the level of cotyledons is the epicotyle, which terminates the plumule or stem tips. The cylindrical portion below the level of cotyledons is hypocotyle that terminates at its lower end in the radicle or root tip. The root tip is covered with the root cap. Embryos of monocotyledons possess only one cotyledon. In grass family, the cotyledon is called scutellum, that is situated towards one side of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of the attachment of the scutellum is epicotyle. Epicotyle has a shoot apex and a few leaf perimordia enclosed in a hollow fluid structures, the coleoptile. Soak a few seeds in water, then split the seeds and observe the various parts of the embryo and the seed. In angiosperm, the seed is final product of the sexual reproduction. It is often described as the fertilized ovule. Seed is formed inside fruit. A seed is typically consists of a coat, cotyledons, and the embryo axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to storage of food reserves. Mature seeds may be non-albuminous, ex-albuminous. Non-albuminous seeds have a no residual endosperm as it completely consumed during the embryo development. A pea, groundnut. Albuminous seed retains a part of angiosperm as it is not completely used up during embryo development. Wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of the nucellus also persistent. The residual persistent nucellus is the periosperm. Integuments of the ovules harden as tough protective seed coats. The micropyle remains as the small pod in the seed coats that facilitate entry of the oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seed become relatively dry, 10 to 15% the moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, they germinate. As ovules mature into the seeds, the ovary develops into a fruit. That is, the transformation of the ovules into seeds and ovary into the fruits proceed simultaneously. The walls of the ovary develops into the wall of the fruit called pericarp. The fruit may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard, etc. Many fruits have evolved mechanism for dispersal of the seed. Recall the classification of the fruits and their dispersal mechanism that you have studied as in earlier classes. In most plants, by the time of fruit development and the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to the fruit formation. Such fruits are called false fruit. The most fruits, however, develop only from the ovary and are called true fruit. Although in most of the species fruits are result of the fertilization, there are a few species in which fruit develops without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpic can be induced through the application of growth hormones, and such fruits are seedless. Seeds offers several advantages to angiosperm. Firstly, since reproductive process as in pollination and fertilization are independent of water, and seed formation is more dependable. Also, seed have to better adaptive strategy for dispersal or to new habitats and help the species to colonize in other areas as they have sufficient food reserve. Young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of the sexual reproduction, they generate a new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for the storage of seeds, which can be used as a food throughout the year and also to raise crop in the next season. How long do these seed remains alive after they are dispersed? This period again varies greatly. In a few species of the seeds lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is the Lupinus erecticus, excavated from Arctic tundra. The seed germinated and flowered after the estimated records of 10,000 years of dormancy. A recent records of the 2,000 years viable seed is one of the date palm. Phoenix dactylifera, discovered during the archaeological excavation at King Herod's palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following question: how many eggs are present in embryo sac? How many embryo sacs are present in ovules? How many ovules are present in ovary? How many ovaries are present in a typical flower? And how many flowers are present in a tree? And so on. So these are the questions, line of questions which you can answer. Apomixis and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants such as some species of asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixis. What is a fruit production without fertilization called? Thus, apomixis is a form of sexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cells is formed without reduction division and develops into the embryo without fertilization. Most or more often, as in the many citrus and mango varieties of the some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into the embryos. In such a species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds, that is observation. Hybrid varieties of several of our food and vegetables crop are being extensively cultivated. Cultivation of hybrids have tremendous increased productivity. One of the problem of the hybrid is that the hybrid seeds have to be produced every year. If the seeds collected from the hybrids are sown in the plants in the progeny, you will segregate and do not maintain the hybrid characters. Production of the hybrid seed is costly, and hence our cost of the hybrid seed become too expensive for the farmers. If these hybrids are made to be mixed, there is no segregation of the characters in the hybrid progeny, then the farmers can keep on using the hybrid seeds to raise the new crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, active research is going on and in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. So that's it. Thank you so much.